Okay, everybody, welcome back to Immunology Biology 466 at Chico State. Um, and for those of you just tuning in to learn a little bit more about the immune system, awesome. Today we are going to keep talking about elements of the immune system and how those elements help defend your body against external insult. And today we're really going to be focusing on the cells and the organs of the immune system. So this is a really good kind of introduction to people who may not know about, all the different parts of the immune system. So first, let's talk about where do all these immune cells come from? Well, all these immune cells come from these cells that we call hematopoietic stem cells, all right? In adult mammals, the main site of hematopoiesis is the bone marrow. You can see here on this diagram, you can basically see down here on the x-axis, it's time and hematopoietic activity over here. We won't go into this too much, but there's a lot of developmental hematopoiesis that goes on in the yolk sac. Then that comes up in the fetal liver and the spleen. That basically goes down as you are born, and then it kind of shifts to the bone marrow. This maintains you for the rest of your life. This is a really interesting topic. You can imagine you have these hematopoietic stem cells. They need to turn into all these other different blood cell types and immune cell types. And that needs to happen for your whole life, right? So if that gets dysregulated, you're either going to get cancer or you're going to have issues where you're not making enough blood to sustain you. So let's talk about the different cells. So your book goes into this, shows you some kind of good pictures. I think it's really interesting to look at these, look at the morphology. I want everybody to realize that that's one of the reasons that blood is a really well-studied system, is because the cells are so easy to get, right? You can just go into a vein, get a bunch of these blood cells, most of them. And they have unique morphologies, right? So they have very different looks. You can definitely discern. Here we have a small lymphocyte. These guys are basically going to be the B cells or the T cells. Hopefully you can see that there's a high nuclear to cytoplasmic ratio, right? The nucleus is very large compared to the cell. Over here we've got a plasma cell. This is basically a differentiated B cell that secretes antibodies. You can't see too much in the stain over here, but these guys are loaded with Golgi apparatus, and basically because they're making antibodies. They're just antibody factories. Now over here on the top right, this is a dendritic cell. Of course, it's hard to get a native picture of a dendritic cell because dendritic cells are sitting in tissue. But dendritic cells are super, super important for the activation of T cells and the initiation of that adaptive immune response. Down here we have my least favorite cell, the mast cell. Hopefully you can see in the morphology, these guys are just packed with all these granules. And these granules are usually containing histamine and other active agents. The reason I say they're my least favorite is these guys are the ones that are really important and related for seasonal allergies, right? So these are the cells that are constantly dumping histamine to cause you to sneeze and have watery eyes and all of those symptoms that you associate with seasonal allergies. Okay, so these are 4 of them. Let's look at some more. Over here we have natural killer cells. Natural killer cells are awesome. These guys are part of the innate immune system, and they can kill cells infected with certain types of viruses. Down below you have a neutrophil. These are important for phagocytosis and killing of microorganisms. You can see this is a human version. It has the 3-lobed nuclei over here. The nuclei always kind of has this lobular shape throughout evolution, but the kind of shape and size and number of lobes changes. So it's different in mice, it's different in zebrafish, but you'll always see the multi-lobed nuclei for neutrophils. Over here we have monocytes. Monocytes are basically circulating precursors of macrophages, and hopefully you can see down below the macrophage looks a little similar to the monocyte. They're a little larger. They usually have these large vacuoles. This is where they take up things and basically gobble them up. We call that phagocytosis, so that's the killing of microorganisms through basically eating them. And of course, macrophages, we'll talk more and more about this, but they're important for activating T cells, and they're activating immune responses. Okay, so moving on, we've got eosinophils. These guys are really cool, again, loaded with granules here. They usually have this bilobed nucleus, and they're really important for killing antibody-coated parasites and basically do this by dumping out their granule contents. Basophils are similar. They're granulocytes down here. They're important for controlling immune responses to parasites. Over here, top right, I don't like this picture too much, this morphological picture, but this is supposed to be a megakaryocyte. Megakaryocytes are awesome cells. They are huge. So these, again, look at these, take these with a grain of salt. They're not all to scale, right? So they just kind of zoomed in on these cells. So megakaryocyte is much larger than an eosinophil, and an eosinophil is much larger than a red blood cell. But megakaryocytes are really cool. They basically replicate all their DNA. They make a huge amount of copies of genes in their nucleus. And then what they do is they just basically crank out proteins, all right? And these proteins go out to the periphery of the cell, and the megakaryocyte eventually just kind of explodes. And that explosion generates platelets, okay? So platelets are actually, if you want to think about it, cell debris, right? They're just chunks of megakaryocytes that are sent out to protect your body against basically bleeding to death. And down here we have a picture of erythrocyte. We always think of erythrocytes as being nice and little red round balls, but when you look at the morphology of them, they kind of look like a donut, and that's because of this biconcave shape. They're kind of like a donut, but with like a membrane in the middle there through the donut hole, which helps for oxygen binding. So we could look at this in a really simplified schematic over here. You can see we've got the hematopoietic stem cell at the top. Again, these are adult stem cells that you're born with, you live with your whole life, and basically they are important for differentiating into all these other different subtypes. In general, we kind of break this down into the lymphoid precursors, and the lymphoid cells would be plasma cells, T cells, NK cells, B cells. These common myeloid precursors over here, they're going to either break off and make megakaryocytes and erythroid cells, so these are the megakaryocyte erythroid progenitor, and they also break off into more of the granulocytes. Now, I should mention that all of these arrows are kind of presumptive. A lot of this is constantly changing as we figure out where do these B cells come from, where do these T cells come from. It's not a direct linear relationship per se between some of these precursors. This is all still an active area of research. So again, these are the innate and the adaptive immune system that are comprising your whole body. So let's think about this. So let's do a clicker question real quick. Which of the following pairs is mismatched? Okay, lymphocytes with innate immune response, natural killer cells with killing virus-infected cells, macrophage with phagocytosis and killing of microorganisms, erythrocyte with oxygen transport, or eosinophil defense against parasites. I think probably the best answer for this one is lymphocytes with innate immune response. And I want you to remember that these lymphocytes are really involved in the adaptive immune response, not necessarily the innate immune response, although, of course, you need an innate immune response to get an adaptive immune response. If you need a little review of that, go back to part 1 of this class and see the link between those. Now, again, all of these guys derive from hematopoietic stem cells, but I do want to give you an idea of, how prevalent they are, right? So this is the proportion in normal blood. So this is not if you have an infection. Obviously, if you have an infection, these numbers are going to change, right? Because your body's going to respond and make more. Let's say you have a bacterial infection, you're going to make more neutrophils. Let's say you have a parasite infection, you're going to make more eosinophils or basophils. But these are, in general, the numbers you should be looking at if you get like a normal cell blood count, so CBC. So proportional leukocytes, and again, this is proportional leukocytes, right? So imagine this is not the proportion of your blood, okay? Because the proportion of your blood is what? The majority of it is red blood cells, right? This is when we're just looking at the white blood cells. So 40 to 75% of those white blood cells are going to be neutrophils. Much less, 1 to 6% are going to be eos, less than 1% are going to be basos. Monocytes, again, these macrophage precursors that are floating around, 2 to 10%, and lymphocytes are 20 to 50%. Again, if you have disruptions of this, if you have an infection, these things are going to change. But if you're just at like normal, static, homeostatic state, and these numbers are off, then you have a problem with blood production. So let's look at what is happening with these cells, okay, and where these cells live. I think this is kind of a cool diagram because it shows basically where your neutrophils are mostly living, okay, which is in the bone marrow and in circulation, okay. Now let's say you get an infection. Let's say you have some tissue, you've got some bacteria, it broke through into the donut, okay. Now what we need to do is we need to get those neutrophils into the region to try to start gobbling up and destroying that bacteria, okay. Now, this is what's happening, right? Remember, we were talking about inflammation before. We've got opening of the blood vessels. We've got neutrophils leaving the blood vessels, right? Because they're normally just in the blood. They're coming over here. They're gobbling up these bacteria, okay? And then the neutrophils are actually going to die. So the neutrophils gobble these guys up. They die. They're engulfed and degraded by the macrophages. And this is what happens when you have a wound and there's pus, right? So the pus is basically dead neutrophils that have gobbled up bacteria and are dying. I want to give you a little information here, which is that neutrophils are short-lived, okay? They don't live very long in your system. They come over, they kill things, and they die. Macrophages are longer-lived, okay? And macrophages also have a unique property, which is they can send warning signals to other cells about the infection. And that's not shown in this diagram here, but the macrophages are basically saying, okay, we've got an infection here. We're going to send out some signals. We call those protein signals cytokines. And those cytokines are going to tell other cells to come over and help fight the infection. Now, one thing that's missing in this figure are dendritic cells. Okay, dendritic cells, as we mentioned a couple slides back, are really important for activating the adaptive immune response. They're like macrophages. Your book talks about this a little bit. Your book kind of differentiates this like they're like macrophages, but they can travel to the lymph system to alert others. I would also argue that macrophages can do that. I think there's some evidence that shows that macrophages can also travel to the lymph system to activate B and T cells. We'll kind of think about dendritic cells and macrophages, I think, for the purpose of this class and for the purpose of really understanding the immunological system, as they're pretty equivalent. They may come from different places, they have slightly different roles, but let's think about them as long-lived in the tissue, and they can send out cytokines, and they can trigger other immune responses besides just phagocytosis. So let's look at phagocytosis and macrophages and kind of how they do this. I like this diagram because it shows this bacteria is coming over. It's got some proteins on its surface. And of course, your macrophage has some receptors that are going to respond to those proteins. And so what it's going to do is it's going to bind them, okay? So these are just phagocytic receptors on macrophages. They're going to bring them in. Basically, this begins to be engulfed. You can see that the cell membrane is going to close around here, make this thing, which we call a phagosome, okay? And the phagosome is going to be loaded with low pH. It's going to become this thing called the phagolysosome, and basically you've got a degraded bacteria at this point in time. So the macrophage has a receptor on its surface for a bacteria. It finds that bacteria, brings it in, and it basically destroys it. Now, macrophages can also do other things, and over here on the right is one of those things that your book is showing. You've got a bacteria. Maybe this bacteria has some bacterial components that are floating around, or maybe it's just the bacteria in general. One of the important things that the macrophage can also do is it can start transcription. And it's not just starting transcription of random genes, but one of the things that it's going to do is it's going to start transcription and translation of inflammatory cytokines. And those cytokines are going to be sent out, as I mentioned, to basically alert other cells that there's an infection and basically activate the rest of the immune system, right? So the neutrophils will come over and they'll just eat a bacteria up and die, okay? But the macrophages are going to eat up the bacteria. They're going to respond to the bacteria. They're still going to eat them up and they're still going to kill them, but they're also going to send signals to other people to say, hey, look, we've got a raging infection over here. Let's help us out. And also, let's try to build, potentially, a memory of what these bacteria look like so that we can fight them off at another point in time. Now, we're going to talk a little bit about the adaptive immune system and why the adaptive immune system is so awesome. So let's go into that. Of course, we'll get into this more in depth later on in the class, but I want you to realize that B and T cells have these cell surface receptors for pathogens, okay? Each B cell has a single type of BCR, and BCR is just short for B cell receptor, and each T cell has a single type of TCR, which means T cell receptor, okay? And these receptors bind to antigen. The key here, all right, is that these are very specific proteins, okay? So in general, immunity against a specific pathogen doesn't confer immunity to another, okay, unless they're really, really closely related, okay? So I've already violated this statement in the first class because we were talking about smallpox and cowpox. It just so happens that smallpox and cowpox have very, very similar antigens on their surface, so the BCRs or the TCRs of your body can respond to both of them. But that is not usually the case, okay? Usually the way it works is a B cell is specific for one type of antigen and a T cell is specific for one type of antigen, okay? And that's basically how they work. So we talked about the clonal selection the other day, and this is kind of the concept behind that clonal selection. You have a BCR that's activated, you get the selection of a B cell that's going to respond against that antigen. So let's look at the structure of some of these proteins. And here is what they look like, okay? So over here on the left, we've got a surface immunoglobulin or a B cell receptor. So this is a BCR, all right? And what I want to draw your attention to is the tip of this finger here, all right? The tip of this finger is the antigen binding site. That's what's going to bind the antigen. All right. Now, the difference between a B cell receptor over here on the left and an antibody is not much. It's basically just this transmembrane region. Okay. So the BCR you could think of as being an antibody that's anchored into the plasma membrane. All right. Now, I like the diagram over here on the far right too because it shows the T cell receptor. All right. T cell receptor has a transmembrane region just like the B cell receptor. Okay. And I just want you to know that T cell receptors are always membrane bound, okay? There is never a free T cell receptor. But look at this. The T cell receptor looks very similar to an antibody, basically one of the arms of an antibody, okay? Now, on the end of these, they've colored them here in red for the B cell receptor, and the antibody is the variable region, and the yellow is basically the variable region for the T cell receptor. And then there's a constant region, and that is blue over here in the antibody and green over here in the T cell receptor, all right? Now, B cells are going to differentiate into plasma cells, and those plasma cells make antibodies, okay? And T cells, there's a lot of different functions of T cells. We'll talk about them as we get deeper into class. But there are cytotoxic T cells, helper T cells, and regulatory T cells. Now, what do antibodies do? Okay, well, antibodies can bind to pathogens, and they can either cause inactivation or destruction, okay? You may have heard of humoral immunity, okay? Antibodies are involved in humoral immunity, okay? And the antibodies, again, as I mentioned, they can have several roles inside of the body. One of them is just neutralizing things, right? So the antibodies can actually bind to pathogens or toxins and basically block them from having any kind of response in the immune system. But they can also facilitate engulfment and destruction of pathogens. And your book has a good description of this on the next slide, so let's look at that. Over here, you can see on the top, we've got these specific antibodies. We've got yellow, blue, and green. Okay, and on the left, we've got, okay, these yellow ones are specific for bacterial toxins. Now, obviously, your cell does not want to receive these bacterial toxins, okay? And so these antibodies, when they get secreted, they basically neutralize them. And what you can see here is the antibodies are all bound to these red circles, which are the bacterial toxins. And then your body has receptors on the surface for those antibodies, brings it in, and they can basically destroy them and break down the bacterial toxins. Now, over here on the right, we've got a slightly different situation that's going on. And we've got like maybe some bacteria in the extracellular space. And what we're doing is we're opsonizing. And opsonizing is basically just labeling these things. And in this case, we're opsonizing them with antibodies, okay? So in this left panel, we've got the blue antibodies, which are kind of binding to the bacterial surface, okay? On the right, we have antibodies over here in green plus the complement system. We'll talk more about the complement system. It's a pretty complicated system, part of the innate immune system, but we will get into that in depth in the next couple chapters. Now, Over here, of course, these antibodies are going to allow ingestion and destruction by the phagocytes, okay? And so the middle one and the right one, it's slightly different ways that this happens, but the end result is the same. The bacteria is still brought into a phagosome, it's turned into a phagolysosome, and basically destroyed. So where do all these lymphocytes live? I've kind of boxed these as kind of some of the important things here, and I've also put a star next to the primary ones where the cells are actually made. So the lymphatic and immune system has a lot of compartments. Again, I think the boxed ones are kind of the most important. So the adenoids, the tonsils, this blocking things that are coming in through the top of your donut hole. You've got lymph nodes where the lymph is getting filtered. You've got your appendix, which is super important. You've got your thymus. This is important for making T cells. That's why the star is next to it. We've also got the spleen, the Peyer's patch, and the small intestine. Peyer's patches and the small intestines are basically lymph nodes for your intestinal system. The spleen is super important for actually filtering your blood. It's not hooked up to the lymphatic system, just hooked up to the blood system. But we've also got bone marrow, and as I mentioned before, the bone marrow is your primary site of hematopoiesis and where your hematopoietic stem cells live. So if you look at the lymph system, it's pretty cool. The lymph system collects the plasma that leaks out of the blood and it returns it to the blood. Okay, so the flow is slow. You don't have a pump, it's not hooked up to the heart, and this is the reason you actually need to move around, move your body. So one-way valves are directing that lymph away from the peripheral tissues to basically recirculate it. So you may have heard that old wives' tale, which is like, oh, if you've got a cold, like just sweat it out, just run it out, right? Get moving, get things, get your body moving. Don't just lay there in bed. It's actually completely true. And the reason is that you're basically going to circulate all of the immune cells through the lymphatic system, and the lymphatic system is going to be able to more effectively launch a response against whatever pathogens are actually inside of you. Now, most of those lymphocytes are really present in specialized lymphoid tissues. This is a terrible diagram, I think, but I guess it's pretty simplified and we can understand it in this way. So we've got the heart, obviously the pump of the circulatory system. It's moving these lymphatic cells. They can go into a lymph node. Again, this is not to size. This is humongous, but lymph nodes are quite small. Those cells can be activated or do different things. We'll talk about what happens in the lymph nodes. They can either go out through the afferent lymphatics, and then, of course, you've got the blood coming out through the veins. So B and T cells can travel throughout the body in the blood and lymph, all right? Not all blood cells can do this, and this is the difference between lymphocytes and the other cells, okay? So lymphocytes are the ones that can travel around through the lymphatic system and through the blood system, okay? The lymph cells in this lymph node, they're constantly in flux, they're constantly coming and going, and the concept is that those guys are going out and patrolling the body. But the majority of the lymph cells are in the lymph nodes at any given time. You don't have a ton of these guys circulating throughout your body. You can go back to that percentage chart that I showed you before, just to show you what's the percent of the blood that is circulating that are lymphocytes. It's maybe 50%. Again, not percent of the blood, but percent of actual leukocytes, so percent of white blood cells that are circulating. Now, we've also got this diagram, which I think is of value so that you can see basically a really simplified version of what's happening when you get an infection, okay? So let's say we're down here, we've stepped on a nail at our foot, we've got the infected peripheral tissue, okay? Those pathogens for the site of infection are going to reach the lymph nodes via the lymphatics, okay? So they're going to basically go to the closest lymph node. So that's how you can always tell where an infection is, right? You can always tell where an infection is because it's going to go to the closest lymph node, the closest draining lymph node, all right? Now, that closest draining lymph node is over here. Of course, we've got blood coming, arterial and going back, venous blood, and we've also got cells that are returning back into the blood via the lymphatics to basically clear out this infection. So we mentioned what a lymph node is, what it looks like. They're very tiny, okay? This is an image. It is about 1.2 centimeters long. There are B cells and T cells that are in different areas here. I've kind of highlighted this. We call it the germinal center. That's kind of where the B cells live. You can see this nice round area. Surrounding that is the T cell area. and we call these parts lymphoid follicles. And of course, you have this medullary sinus. Let's look at the diagrammatic version of this on the next slide. This is just a morphological section of a lymph node to show you what it actually looks like. But let's look at the diagram. I think maybe it's easier for us to look at and think about. So we have this afferent lymphatic vesicles. Dendritic cells can come in here, okay? We've got these lymphoid follicles on the edge, mostly B cells there. The germinal centers are also B cells. Then we've got this T cell region, this blue area, okay? And of course, we've got the medullary sinus where all of the cells are coming and going and mixing, all right? So the concept here with the lymph node is pathogens or dendritic cells can come in, okay? So the dendritic cells have already gobbled up a pathogen. They have it on their surface. They're presenting it. And then what they're going to do is stimulate B cell proliferation. And that B cell proliferation is going to be in a germinal center. And then the T cells are going to talk to those B cells and also proliferate. A really important thing here is that the lymph is totally filtered, right? So all of the stuff is basically getting filtered out in this lymph node, all right? And there's not going to be any pathogens that escape from this lymph node, all right? So within this lymph node, these guys are hanging out. Pathogens are not going to get out. They're going to basically be processed and destroyed in the lymph node. Now, this adaptive immunity really happens in these secondary lymphoid tissues. So let's look at this diagram. Again, we've got an infection. Let's say you stepped on a nail, you got an infection. Macrophages are over here. They're gobbling up the red bacteria. They're sending out cytokines. They're telling neutrophils to come and go. But one thing that's important is these dendritic cells, okay, they are now carrying the pathogen on them. So they have stuck some of the bacterial components on their surface. And what they're going to do now is they're going to go and look for help, okay? So they've gotten into the lymph system. They're going to come in here into the lymph node, all right? There are macrophages within the lymph nodes that engulf the bacteria. We've got the dendritic cells are activating T cells here. These T cells are going to activate B cells and produce antibodies. Again, this is a super simplified version of what's happening. We're going to talk about all the different specific molecular mechanisms and how they're controlled in your body in the upcoming chapters associated with this. So we talked a little bit about the spleen. I just want to tell you that the spleen is a very special organ. The spleen functions to filter the blood, okay? One thing it does is processes bloodborne pathogens, okay? But that's in the white pulp. And it also removes red blood cells. It removes the dead red blood cells. RBCs is how I have that abbreviated here. But what's really important is the spleen functions to filter the blood, okay? Your blood gets filtered by the spleen. Here's the spleen. Here's a diagram of it. You can see this white pulp over here, and you can see the red pulp all around it. And if you look at the transverse section of a white pulp, look, it looks very similar to a lymph node, right? We've got the T cell area in blue. We've got the B cell area in yellow, okay? We've got germinal centers. We've got marginal zones. We've got these perifollicular zones. All of these are basically, if you want to think about them, mini lymph nodes that live in the spleen. Again, I don't like to say lymph node on this because it's not really lymph. It's filtering the blood. But you can think of the spleen as basically the lymph node for your blood, okay? It's the filtration system for your blood. So let's do a clicker question real quick. Which of the following is the predominant route by which pathogens are brought in from the site of infection into a lymph node? So do you think it's from the efferent lymphatics with an E, arteries, veins, afferent with an A, lymphatics, or high endothelial venules? I think the best answer here is afferent lymphatics, D, and again, afferent with an A, lymphatics. That's how it comes in. How it goes out is the efferent lymphatics with an E. What we probably don't appreciate is that most of the secondary lymphoid tissue is associated with our guts, okay? So think about this as, remember I told you our bodies are kind of like a big donut, and the top of the donut is the mouth and the bottom is your anus, all right? Remember, this part of your body has the highest residence of microbes, all right? the respiratory system, the gastrointestinal tracts. We need to guard them really, really well, okay? We guard them with all these different tissues. We will get into the specifics of these tissues as we go on in class, but I want to introduce the terms to you. The first one is GALT, gut-associated lymphoid tissues. These are your tonsils, your adenoids, your appendix, and your Peyer's patches. We'll look at Peyer's patches in the next slide. They basically are like little lymph nodes that are right up against your gut, right up against the epithelial cells of your gut. We've also got BALT. These are bronchial associated, so in your respiratory system. And MALT is more general, but this is mucosal associated. Again, we'll go into all of this in more depth, but I want you to know the terms so you're not confused when you're reading or coming across these terms elsewhere. So here is an awesome picture of basically a Peyer's patch. We've got the epithelium, okay, so the gut lumen's up here. This is your gut. You've got these cool cells called M cells. They actually let things through to talk to dendritic cells. And then, of course, in this region, we've got a T cell region, which is blue, and we've got this B cell region, which is yellow, and we've got a germinal center, and these are efferent lymphatics. So things are coming in through the M cell, getting processed by dendritic cells, T cells, and B cells, and going out through the efferent lymphatics. If you want to look at this on a slide version, the actual morphology, it's on the right, just to kind of show you, what the B cell region and this germinal area look like and all the T cell regions surrounding it. But basically, we can think about this as kind of the lymph node of your gut as well. Okay, so let's wrap this up with a final clicker question. So which of the following explains why immunity to influenza may appear to be relatively short-lived? Okay, this is an important one. It gets into COVID, infectious diseases, all these issues. We'll talk more about why this is correct in following slides and following classes. But A, effective immunological memory fails to develop. Okay. Or B, immune responses to influenza involve innate immune mechanisms only. C, the primary and secondary immune responses are equivalent. D, influenza virus targets memory cells. Or E, new influenza variants able to escape previous immunity appear regularly. And the answer to this, as you may or may not know, is E, new influenza variants able to escape previous immunity appear regularly. This is why it's so important for us to have the technology to generate new, novel versions of vaccines as pathogens pop up. Remember, we're not the only thing evolving here. The viruses and bacteria, pathogens, are also evolving with us. And if we can't stop them, then they basically take over and destroy us. If we stop them, then we win. And one of the ways that we can do this is by creating new vaccines that recognize these new variants, training our bodies to fight them off before they get a foothold in whatever population they like to live in, whether it be animals or humans. Okay, so that's it for today. Join me next week probably, for Chapter 2.